double act next, and it's from Andreas, Beyer and Argyris, Papantonis, and they're going to tell us about living in the fast lane, accelerated copying of genetic information with aging, and that will be of particular interest to yours truly and one or two others, no doubt.
Yeah, thank you for the introduction, and I also we thank the two Larrys for inviting us. This has been an amazing meeting and really a unique experience, at least in my scientific life, and probably the same for Argyris. Yeah. So yeah, and it's also very humbling to give the last presentation at this conference. So you feel like now we have to answer all the questions that were raised at the meeting. But of course, that's not going to happen. We will just make it worse. We will add more questions to the list. So sorry for that. So I'm a computational biologist. Larry Hunter is a big guy, a hero in our field. He created some of the most important conferences. And so being invited by him also actually means a lot to me. So thanks. Yeah, so aging is one of the things I'm working on. Also, Argyris is working on many other things than aging. And I started working on aging when I came to Cologne 10 years ago, because then I became a member of this institute, which is working on aging. Actually, at that point, I didn't know anything about aging. They recruited me because I knew something about computers. But then, once I was in Cologne, I realized that aging is really a fascinating field. And so that's what we want to start with, getting you excited about working on aging and the molecular biology. So what you're going to learn today is a new molecular mechanism that contributes to aging. We think we know a lot about aging, but if you then really drill deep and ask, what is really causing aging at the molecular basis, there's not much. We've just heard about telomeres. That's quite well understood. DNA damage is also well understood, relatively well. There's still much to learn, but other than this, we know very little about what is really causing aging. And the aging field has many other interesting questions to address, and I want to introduce you to some of them because it's really fascinating, at least to me. So one big question is, why do we age in the first place? That's absolutely not trivial. So you might think, it's kind of course we have to age, of course we're aging and then we will die, but no, it's not that simple. Yeah? So for instance, there's this Greenland shark, which is getting maybe 250, maybe 500 years old. We don't really know. It's somewhere in this range. But even at the lower end, if it's only 250 years, it's presumed to be the longest living vertebrate. 250 years is way, way longer than any human, obviously, right? So 10, 20 years, what is the maximum lifespan of humans, is not a physical limit. And then, of course, there are organisms that are even much older, like these trees, which get more than several thousand years old. And there are organisms that don't really age at all. We have learned about the planarians yesterday, and hydras are similar in a sense that they can also reproduce in a kind of budding process, and they can completely repair themselves. And they're also thought to essentially not age at all. So not aging is possible. So then again, why do we age? So currently, I think the common sense in the field is the explanation. First, aging is not a physical or chemical necessity. We don't have to age because of some kind of physical or chemical wearing off, but it's much more determined through evolution. So for instance, if you take a mouse, if you would make a mouse that lives for 20 years in the wild, it would be eaten much earlier by some predator. So it doesn't make any sense for a mouse to be able to live 20 years. So then it also doesn't make sense to mount all the molecular machines that you need to live that long. So instead, for mice, it's much better to reproduce as early as they can, before they get eaten by the cat. Okay, as I said already, the other big, or one of the other big questions is, what are the molecular foundations of aging? So how does aging influence cells and organisms? And those are the questions that we are addressing in the work that we are presenting now. We are starting very, very low level because we realize that some of you don't have any molecular biology background. So it's kind of a genetics 101, molecular biology 101. But then we also have some more sophisticated stuff for the experts among you to hopefully make everybody happy, if that's going to work. So genes are of course important, and why are they important? Because they encode the structure of proteins. So if you wish, it's like a construction plan. Each gene is like a construction plan for Lego bricks. And then with those bricks, you can do all kinds of fancy things. And so this is similar like a construction plan for proteins. So genes are construction plans for proteins. And then you want to make cells. And different cell types need different types of proteins. So as in the same way as you need different Lego bricks to make different models, you also need different proteins to make different types of cells. So here we have a neuron and some muscle cells, and they need very different types of proteins. There's a lot of common stuff, but also proteins that make them different. And actually, these differences define the function of different cells. So when we age, two things can go wrong. And until we started this work, the field was mostly focusing on the left-hand side. I'm still getting confused about the buttons, like everybody. So one thing that can go wrong is that you produce the wrong amount of proteins. So that's one thing that clearly can go wrong. But the other thing that was, at least at the level that I'm going to present, kind of ignored is the quality of proteins. So you make the right proteins, but you make them at a poor quality. So you probably all know about protein aggregation as a cause for many, presumed cause for many neurodegenerative diseases. So that clearly is a quality issue and not necessarily a quantity issue. So in our work, we focus on the quality side of things, and not on protein folding, but on the reading of genes, the transcription process. This work is a huge collaborative effort. That's why you have two speakers here, but there should be four. Linda Partridge has been also very important, and Adam and Tabby as really senior contributors to this project. And I mean, all of these different labs are not only important because they were providing a lot of data, but also the intellectual input has been very important already from the very beginning of the project. Maybe this is something for the Q&A part, history of science. And then all of this culminated in this paper that we've published very recently on aging-associated changes in transcription influence longevity. And that's, I think, important because really only during the last year or so, 12 months maybe, this paper and several other papers came out that showed that the fidelity of transcription is declining with age, and that this is really contributing to aging phenotypes. So this is a very new thing. So biology 101. Here we have the genes which are encoded on the DNA, and then we have this process which is called transcription, which is creating copies of genes using RNA. So the RNAs are copies of genes, and then the RNA is translated into proteins, which are the functional machines, which are the important part. So now what we could say expression level, so the amount of RNA that's produced from genes is determined through other proteins that have regulatory functions, which usually bind to DNA, and then, for instance, they change the epigenetic features that Caroline was just presenting to us, and that controls expression levels. So another thing that can go wrong when we age is this, so that the transcription process itself is in some way dysfunctional, or the fidelity is reduced. And if that happens, and this has consequences for the RNA, so the RNA doesn't have the proper sequence maybe, and that would then lead to issues with the proteins and then consequences for the cells. So the question we were asking is, how does RNA biosynthesis itself change during the aging process? So transcription works through polymerases. They were also just introduced by Caroline. You need polymerases to copy DNA, but you also need polymerases, these are other types of polymerases, to make RNA, to create these transcripts. So this is called RNA polymerase II or just Pol2. So that's the machinery that reads the genes. And we were using a way to measure the speed of this transcription process. We did not invent this, but this has been very handy, using relatively standard technology, which is called RNA sequencing. So RNA sequencing is just what the name is saying. You sequence the transcripts, you sequence the RNA. That's very useful for determining the amount of transcripts that you have from a specific gene. And the standard protocol for RNA sequencing is just measuring these exons, which are parts of genes, which are actually then eventually leading to proteins, while genes also contain huge regions, which we call introns, that are actually removed. So you first make the transcript, and then you cut out these introns, that's called splicing, and then this is thrown away and not used afterwards for making the proteins. So it's kind of debris or waste. But for us, it's very useful. If you use a different protocol, which is also sequencing these parts here, these introns, which are afterwards thrown away, you can use this information to quantify transcription speed, elongation speed. So the exonic part is not useful, but the intronic part. And this is why we can't use this standard protocol, but other protocols. Factory sequencing was developed by Argyris, for instance. That works really well. And what we utilize is what you see already here, this slope. So when you create this data, you will always see that in the introns, the redistribution is declining from the beginning of the intron towards the end of the intron. And this steepness, the slope, is a measure for the elongation speed. So here you have this cartoon of an intron. Again, the exons are flanking this intron. And then you have the slopes. And when the slope is steep, then this is corresponding to slow elongation. And when the slope is shallow, you have fast elongation of the polymerase. So here's some real data. This is a gene in flies at young and old flies. So flies get about 50 days or a bit older. So this is 50 days is quite old, and 10 days is young. So here you see the noisy data, but then you can just fit a regression line into this intron. And what you see is that at day 10, you get the black line here. The black line is slightly steeper than the blue line, which is the old fly. So there's a tiny, tiny difference here, which we interpret as an increase in speed because it's more shallow in the older flies. You see all the noise here. So if you have that only for a single gene, that could be pure technical noise. This may have no meaning. So what you really have to do is you have to average such data over entire genomes so that you average over hundreds or thousands of introns. And that's what we did. So I think one distinguishing feature of this work is that we have been using so many different model species, from worms here to flies, mice, rats, and humans, different organs, different types of cells. Again, that's for the Q&A part, why we were using so many different models. And this was also one of the very nice aspects of this interaction of the scientists in Cologne back when we started the project. So here's the data. If you see a circle on this dashed line here in the center, it means that the average elongation speed doesn't change between young and old. If you are on the right-hand side, then the average speed has increased. If you are on the left-hand side, the average speed is reduced. Average is important here. So now the blue dots are all comparisons where we compare old versus young. And all of the blue dots are on the right-hand side, meaning that in all of these models, in all of the conditions that we were testing, the average elongation speed is increasing with age. The orange dots, they correspond to interventions that extend lifespan. Known interventions, which are established for decades, like dietary restriction, for instance, repressing insulin signaling. And for most of those interventions, you see that they reduce the elongation speed. So that's just correlation. Yeah? So the speed is correlated with aging. That doesn't establish causality yet. But a few points which are important. First, I was emphasizing the average elongation speed is increasing. So we usually see hundreds of genes or introns for the experts where the speed is decreasing with age. Yeah, so we have hundreds of genes with decreasing speed, but you also have hundreds or even thousands of genes with increasing. So on average, you have a small increase. Another important point is that the effect sizes are tiny. So the average speed increase is really very small. And for a single gene, this would have no effect whatsoever because the cell have amazing quality control processes, and those processes will be able to handle this. But because hundreds of genes are affected, those small changes are adding up over those many genes and eventually leading to problems. So this experiment here was very important to then establish really the causality of that Pol2 speed changes contribute to aging phenotypes. And in this experiment, we created mutants of worms and flies where the Pol2 elongation speed was reduced. So through genetic mutations, we reduced the speed. And this reduction in speed increased the lifespan of these organisms. So here, this is the wild type worm, and the black line is the wild type fly. And then this yellowish or orange line is the intervention where we reduce Pol2 speed. And they live a little bit longer. It's not much, but it's a significant increase in lifespan. So that now shows really that the elongation speed contributes to, let's say, age-associated phenotypes like longevity. So now we have two big questions, which our paper does not yet fully answer, but we are trying to contribute a little bit. One big question is, what's the issue? So why is it a problem for the cell if the transcription speed is increasing? And the other big question is, what's causing the speed increase? And we're now trying to give you some hints, at least, on those two questions. So one thing that we discover is that if the speed is increased, the polymerase starts to make more mistakes. RNA polymerase has amazing quality control processes. It can even recognize that it made a mistake. It can then backtrack a little bit and then correct the mistake and then continue transcription. So this is really wonderful. But it seems that if it's too fast, then it will, to a tiny amount, but apparently significant, increase the error rate. Second issue, splicing efficiency is changing. Here, I will not explain the details of splicing. That's this thing, where you cut out the introns. That's a change that we observe, which is very consistent, that the splicing efficiency goes up. We don't know yet if this has really functional consequences for the cell. So far, it's just an observation, what's happening. Another observation is that circular RNAs go up. So mRNA is not supposed to be circular, but it's supposed to be linear. Some circular RNAs actually have function, but most of them are presumed to be just mistakes, erroneous consequences of splicing. And they also go up. So here's the data for the first two things. So this is the mismatch. Here we're quantifying the mismatches between these RNA sequencing reads and the reference genome. Many of those mismatches which you observe in the data might be just due to sequencing mistakes, so purely technical. But we would assume that the error rate that you have, this technical error rate, is the same in young and old cells. So that if this mismatch rate goes up, this corresponds to really like an aging-associated change. And you may notice that not all of the blue circles, but most of the blue circles are on the right-hand side, corresponding to an increase in the mismatch rate, while most of the orange circles, these lifespan extending interventions, are on the left-hand side, so they have less mismatches. This is the splicing efficiency. So here we just count how many reads you have that bridge exon-exon junctions. So these transcripts are already spliced. And then we compare this to reads that bridge intron-exon junctions. So here the intron has not yet been removed. So these correspond to unspliced transcripts. And this ratio between these two types of transcripts is actually decreasing. So the splicing efficiency is increasing in the older samples and Yeah, and the lifespan extensions, it doesn't really change. Okay, now Argyris will take over and tell you a little bit about possible causes underlying the speed increase.
That's true. Okay, so thank you from my side as well. It has been great despite the jet lag and so on and so forth. Looking at this, I now understand why everyone has had issues. It is absolutely unintuitive, but okay. So yeah. So this goes all nice and well. We submit the paper, and as is accustomed for those of you that has ever submitted a paper, reviewer number 3 comes in and says, oh, this is absolutely great. Why? And of course, the truth is we didn't really have much of an idea at the time. And what we knew was that chromatin structure, which is what Caroline was talking about, can actually influence how fast the polymerase will translocate along DNA. Because of course, if you think about it in very pragmatic terms, it is an enzyme that has to open up the DNA double helix, remove all the proteins or the nucleosomes that this is wrapped around, and transcription factors that are bound along the way, and so on and so forth, to make the copy. So this requires some sort of an effort. So the fewer obstacles along the way would presumably allow this to happen at a higher pace. And it was observed way before we did anything on it that actually aged cells have fewer nucleosomes along the DNA fiber, or let's say more loosely positioned. So Caroline just showed a very similar one. So DNA is not naked inside the nuclei of human cells. It's actually wrapped around histones, so I don't have to reiterate any of that again. And then it goes all the way up to forming chromosomes like the ones were being shown when we are in high school. And what we knew, for a completely different project at the time actually, is that when human cells enter a state of cellular aging, they actually have significantly less histones inside their nuclei. So this would mean that there's fewer histones available to wrap the DNA around, and this would cause something like this. So again, it is not absolutely understood how and where these histones are positioned. But we do know that by and large, they occupy, in a young cell where things are nice and jolly, they occupy positions that are more or less well-defined. So if you were to map them, you would find multiple nucleosomes at the same position, more or less. So it's pretty sharp, and they're pretty close together. They form pretty canonical arrays for the most part, and so on and so forth. And then in the old cell, you actually have a much more fuzzier positioning. A, because you have fewer of these, so you have a landscape to populate with fewer proteins, and B, for reasons that we don't understand at all. But it's simply fuzzy. So a given position will actually now be represented by overlapping signals from nucleosomes that are not exactly at the same position. So when we looked at this, and this all was done in human cells, we actually find that indeed the density goes down. And it doesn't go down across all features, but again, on average, we have a lower density. This would mean that the polymerase has fewer obstacles to work against. Now, if this is the case, you could put back the histones that the cell is missing, restore perhaps the canonical structure of histones along the chromatin fiber, and this would slow the polymerase down. And if the polymerase being slowed down is at all important, this would delay the onset of aging. I've done this experiment with my own hands. I'm really, really proud of it. It's one of the very few that I'm allowed to do in the lab anymore. And I honestly, the reason I did it, because I didn't dare give it to a student, because I thought, I told Andreas at the time, we can try it. I'm not sure this is going to work. It did. So we overexpressed H3 and H4. We forced it upon these human cells. And actually, when we measured the speed, we saw that it did reduce the speed by which the polymerase would translocate along gene bodies. And to add insult to injury to my expectations, especially this construct, H3, and I'm happy to discuss afterwards why this one way more than the other, actually delayed the onset of aging or senescence of these human cells. Again, for the field of aging, the senescence of human cells in vitro is really not that convincing. It's not organism aging per se. But the lab of Lindra Partridge had a very similar experiment where they were overexpressing histone H3 in Drosophila cells, actually in the glia in the brain. And that alone was enough to allow the flies to live longer. So this corroborates the idea that if you effectively slow down the polymerase, you can extend lifespan, and you can do that by replenishing the chromatin landscape of a given cell type. So the current model would be that aging manifests through changes to chromatin, for which we don't have an explanation at the time. So why? Is it a choice that is deterministic? Is it a program, or is it stochastic? I know, Mike, yes. I agree. So, or is it a stochastic deterioration of chromosomes? And this would, of course, allow an increase of transcriptional elongation. Again, probably stochastically, because now there's fewer obstacles, so the polymerase has no other option but to start going faster. This would then lead to splicing and transcription errors that would then carry over to translation and formation of proteins. You would have a landscape of proteins that are not fully functional or execute their functions with less fidelity. And this impacts lifespan of a given cell, tissue, or organism. Now, if you take a step back, you can, of course, say, well, this means that if we slow down Pol2 in human cells, we would allow the whole human, from head to toe, to actually live longer. This is not absolutely true because, as a small parenthesis, many of these interventions that really work wonders with lifespan actually have side effects. Would we want to do that then? Are we willing to cope with that? Also, do we really want humans, and this is semi-philosophical, to live longer? Maybe the idea would be that we live exactly the same, which is, fair enough, but have higher quality of life. So health span versus lifespan, which is also a very big discussion right now. Would that extend lifespan? And what would be benefits or non-benefits of having such intervention? And yes, would we want to live forever? So I don't know, you tell me. So acknowledgements, this is the group of Andreas in Cologne, and this is Papantonis, the other Greek connection, who, almost single-handedly done all the revisions, all the analysis. It was a very painstaking process. So the truth is that we owe much to him. And this is Sia in my lab, who, except from the one amazing experiment that I'm really proud of, did much of the other work for the revision. And of course, the labs of Adam and Lindra and Thomas that provided worms, flies, mice, and all the input that Andreas already discussed. And I guess with that, we can stop and take questions.
Thank you very much, both of you, for that provocative presentation. So the work that you described latterly was the work that was done in between the sending of the paper in for review and the publication date of 2023, because, whatever it was, I noticed there was a 2.5-year gap between the receipt and when it was published, which is kind of unusual, but I'm glad you got it published. Anyway, just that I assume was the work that was in between, right?
Yeah, it was a very long process.
Okay, Tom, you got a question?
Just to answer your last question, yes, we do want to extend lifespan. I actually have a question, too. Do you know whether the speed of polymerase is changing because it's just changing the rate at which it's putting in bases, or whether it's altering whether it sees pause sites, how long it spends at pause sites, how many of the pause sites it stops at?
We don't. Yeah, what we see is an average over millions or, I don't know, even billions of cells. Is it turned on? Yeah, I think it's on. So we don't know the answer because we are averaging over millions of cells in any sample, and yeah, then we have no information on where the polymerase is pausing.
Okay.
We will be doing the chip to see where it might pause and whether this changes, but
Yeah, I mean, there's this, okay, for the other, you probably know, based on your question, I think you know. So there's this promoter proximal pausing. So the polymerase is starting transcription at the beginning of the gene, and then it stops for a while, and then it keeps going. So that is definitely not explaining these phenomena. So this is not related to this promoter proximal pausing. But there could be other, more stochastic, not random, pausing in the gene bodies. We don't know.
Because you might have very different outcomes if it's really due to changing the pausing, especially on splicing.
Yeah,
that's the idea.
that's another point.
That's the
yes. Yeah.
So, I'm interested in your thoughts on the signals during the aging process that lead to reduced nucleosome assembly and hence changes in the polymerase rate. And there's two thoughts. Reduced caloric intake in rodents enhances lifespan. That's experiments people have completed. Also, lipid peroxidation reactions impact mitochondrial function and create cellular damage. Are those processes that indirectly could be impacting nucleosome assembly and hence polymerase rate? Any experiments done, correlations there? I can think of organisms that have high metabolic rates, that have long lifespans. What's been done to link those together, to get a handle on what drives what?
Yeah, there are many chemical modifications that possibly contribute to aging, like reactive oxygen species, ROS, but I don't know of such data. Do you know?
So there is, with the exception of damage actively causing the eviction of nucleosomes around damaged sites in order to facilitate repair, there has never been a systematic, as far as I know, address of whether having, for example, high ROS would make a landscape that is less susceptible to incorporating nucleosomes. I don't know that.
But there are mitochondrial mutations that lead to reduced lifespan. I think they happen in humans as well as other organisms. Have you looked with such events at what happens with your chain of reactions?
No.
Thank you.
So Jess Tyler showed a while ago that this phenomenon of histone depletion in old yeast cells exists, and so this seems to be like a general phenomenon. But then more pertinent to your talk, David Bentley, also a long time ago, constructed some polymerase mutants that are either faster or slower, and so these might be really fun to test, just whether it's speed or just histones.
Yes, so yeah, of course we're citing Jessica Tyler's work. We had her visiting us in Cologne, and that was a very important inspiration for this explanation. Yes, and I mean, we were using these mutations that slow down POL2. But as far as I know, if you put them in mammals, they are embryonically lethal. So it's not a simple story. But at least in worms and flies, it works. It's extending lifespan.
Yes, I have a question. First of all, I want to congratulate you not only for the work, but for resisting 3 years of revisions. That's something. My point is, from your experiment, ultimately, I do not know, but maybe I missed something, whether it is the ratio of histone to DNA that prolongs the life, or the speed. I understand that there is a correlation between the two, but I don't know which one is the critical event.
So let me put it this way. So the fact that you have fewer histones, in my mind, will, whether you like it or not, allow the polymerase to go faster. So if you had to hierarchically put them one before the other, you would say that the loss of histones and the fuzzier landscape allows the polymerase to go faster, that leads to errors, that leads to The question is whether, again, whether this loss, this fuzzier landscape, is actually causally related to initial signals of aging or telomere attrition or whatever. We do not know that. So it's difficult to say, but it has to be the mistakes from the polymerase that translate into proteins or faulty transcripts that have the functional effects. It cannot be just having a fuzzy landscape, which I should say at the same time would in theory allow larger stretches of less protected DNA for mutation. So it could be a combination of the two that, again, we haven't really gotten there.
Yeah, I cannot resist. I'm sorry. And has anybody ever said no to you want to extend human lifespan? I mean, I think it's a pretty universal desire, but put that aside for a second.
My grandfather, but I'm happy to discuss afterwards.
Okay.
And he had really good
and
arguments.
health span.
Health span, no. Oh, definitely.
Everybody wants longer
We all
health
want
span.
that, yes.
That's a different thing.
Yes,
Yeah,
that's a different thing.
So I'm curious about your comment about the possible side effects. Obviously, a problem. We just saw a paper about extending telomere length can really be bad for you. So obviously, important question. You did this H3 overexpression experiment. Did you notice any phenotypes other than longevity? Were there side effects of increased H3 expression?
Well, there was the H4 experiment, right? And there, we actually observed a very strong decrease in speed. And these cells did not really delay going into senescence. If anything, they had a marginal advantage, which was very, very little compared to controls. This either means that H4 is not the right histone to do this job. I should say here that the H3 that we overexpressed is the 3.1, so it's the replication-dependent histone, whereas the H4 is not. So it was either that, or the fact that if you overdo it, so if you slow down the polymerase too much by, I don't know, making a far denser fiber, you might have detrimental consequences. And this is how Andreas actually, when I sent him the data, this was his immediate response. You probably overdone it with the H4.
I have a question. So if I were a pharmaceutical company as opposed to a food company,
Okay.
if I was a food company, I would be horrified at the idea of caloric restriction as a way of living longer. But if I was a pharmaceutical company and I had an elongation slower, I mean, there are many, and if you're not careful, of course, and you drive the rate to zero, you would die. But there must be doses imagined by pharma companies to do this. And so I guess the question is not what are you doing about that, but have they all been calling you to help them with their efforts?
Have you gotten any calls? I haven't. Oh, you have?
Yeah,
So
yeah.
Andreas is becoming rich and I'm not, I think. This is the bottom line, I guess.
There's no correlation between calls and richness, I have to tell you.
Fair enough.
I wonder if the following explanation for the consequence of high speed of transcription makes sense. So when the rate of transcription is nice and slow, the transcript as it emerges, has time to fold into a structure without interference from RNA that's yet to be transcribed. And when you race through, you spit it all out and it can fold in a way that's quite stable, and yet then it affects the rate of translation, which could affect the folding of the protein. Is that something that's worth adding to your list of why, what is the problem?
I mean, the folding of the RNA is definitely important for the splicing. So I think that the speed increase has consequences for the splicing, but that is absolutely not trivial. Others have already published that increasing elongation speed can cause intron retention, exon skipping, all kinds of different changes in splicing, which may in part be related to the RNA folding that you were mentioning, in part being related to proteins that decorate the transcript, the nascent transcript, to regulate the splicing and so on. So this is creating such a huge complexity that it's very difficult to really, explain what is the consequence of the elongation speed and what is just response of the cell to the stress.
Great. Well, look, thanks everybody. Thank you for the attention. Thank you two guys for a great talk. Meredith is going to say a few words to wrap things up with Larry H, that is. In the meantime, I just want to say something that I think the 3 E's are represented here, and that is it's educational, it's exciting, and it really has an energy to it, which is very unusual. And I suppose there's a 4th one. It's eclectic. It's a very eclectic set of talks, and I always enjoy coming, and I thank you all for coming too. So, Meredith and Larry, over to you.
I just wanted to start because Larry gave me such a lovely shout out this morning that I wanted to make sure that we also recognized Larry Hunter for his wonderful contributions over the last few years. So thank you so much, Larry Hunter. As Larry Gold alluded to in his letter, we've had the wonderful privilege of being able to have weekly calls with Larry Hunter for almost the past 2 years. It's been pretty wonderful. And I think, our book club is something that I hope we can continue. And I do believe that having you in our lives makes us better. So Larry Gold, thank you so much for bringing Larry Hunter into my life and to this community. And thank you, Larry, for everything you've done.
It's going to make me blush. Thank you. And one of the things I wanted to make sure to do is to thank Tim and Melissa and Ruth, who am I leaving out? The other moderators, Simon, who did a great job. The moderators are really very important for keeping things going and calling on the questions and everything. So thank you very much to the moderators.
I'm coming back because if you're saying that, we have to give a shout out to Digger and his team. So thank you so much to the production team.
Yes, the production team was awesome.
Phenomenal. And to Grace and Emma, who really behind the scenes have made this happen for us. So thank you so much.
Well, and I don't know yet, maybe you can tell me how many people watched online. Do you know?
We had about 800 people join us online. So thank you so much over the course of 2 days.
And do we have a geographic distribution?
I don't have that yet, but I can
Okay.
look.
So thank you very much to the audience who brought great questions, who brought enthusiasm. And I was a little worried because we've never done this live stream before from here. We've done it in a small venue during COVID. And the idea that the auditorium was not quite as full as it was just before COVID. And I was worried that we may have lost people. But apparently, if we had 800 online, we've doubled the size. So thank you all who came and who participated with your brains and your questions and everything else. And I really look forward to seeing you all next year.